So today we're going to talk study designs and protocol development. It's just a short overview. So the very first thing I start with is actually the main objective of the study. And the reason I start with this is because I know we all work with and have worked with investigators who ask us a question, but they have no idea how to actually develop an objective. So I don't know if you have heard the acronym FINER before, but FINER stands for feasible, interesting, novel, ethical, and relevant. And so when you're working with an investigator and they throw out an idea, something that they want to evaluate, we can use the FINER principle to help them narrow down to what their true objectives are. Just want to throw that in at first. So let's talk some research study designs. So there are multiple types of research. So you have your basic research, typically descriptive in nature, used to understand and explain phenomenon. We want to generate new ideas, principles, theories. With basic research, it may not be immediately utilized, but we're starting to form that basis. Translational, of course, is your bench to bedside. We don't do a lot of translational work at Cook. We have one investigator who has done a little bit of translational in working with UNT Health Science Center. But for the most part, that's not where we live. Applied research is finding solutions to everyday problems, illnesses, developing innovative technologies. So here we're going to examine a specific set of circumstances, and we want to look at real-world application. We do a little bit of applied research here at Cook, not a lot, but we do some applied research for sure. And then really kind of the bread and butter of at least our research operations is in our clinical research. So here we're evaluating the safety and efficacy of medications. We're looking at devices, diagnostic products, treatment regimens. Here we have an entire bibliography of drug device biologics that fall under clinical research. We also sometimes tie our observational and expanded access work into clinical research, because even if it's observational, even if it's retrospective, we're typically looking at the effects of medications, diagnostic procedures, and such. Okay, you guys probably have seen this schematic or know this schematic. It's just kind of a general overview for different types of study methods. So as you know, a lot of what we do is observational. So we're studying a phenomenon, but we're not altering in any way what's happening. So here we're looking at our cohort studies, we're looking at our case control studies, and our cross-sectional studies. We do a lot of observational work, retrospective and prospective, in the work that we do. We also do some interventional, and a lot of the talk today is really going to kind of focus more on the interventional, because we're starting to do more of that. And it's a different type of protocol. It's a different type of study design. So with interventional, of course, we're intervening to change reality in some way and then observe what happens. So here we have our quasi-experimental, our adaptive clinical trials, and our randomized controlled trials. So there's three main approaches to research. What we do at Cook is almost 100% quantitative. So we're looking at correlational, causal comparative, experimental, quasi-experimental, survey design. We have some qualitative work, especially with the medical students. We do lots of case studies. We've worked with chaplains who've done a little bit of grounded theory, ethnography. There's phenomenological research, narrative research. We don't do a lot of that. We're starting to look with some of the areas that we're working in at doing a little more qualitative work, but really predominantly we're steady in the quantitative space. There's also a mixed methods model. So you're taking a pragmatic approach to research, and here you're looking at some sequential, some concurrent, some transformative research. I'm going to say that I don't think we may have one or two studies that have used mixed methods, but like I said, we don't do a lot of qualitative. And I know for a fact we've done some that have mixed methods, because we have some focus groups and other type of studies that have mixed with some quantitative approaches as well. All right, let's hop directly into protocol development. So First question, get this all the time. Do we need a protocol for everything we submit to the IRB? And the answer is 100% affirmatively, yes. IRB will kick it back. It doesn't matter how small our data set is. If we're only doing a retrospective cohort, they want to see a protocol. So I'm going to transition, like I said, into really talking about interventional studies. When we're developing research studies that are really more than retrospective, we want to develop a synopsis and a schedule of events early in the process. And the reason being is that our protocol synopsis is really an overview or a summary of the protocol. We're having those conversations and we draft an outline of what we think that our study should look like. We can go ahead and write up that protocol synopsis. The other thing that we can do is start looking very early on at our schedule of events. So this is actually a table of all of the study-related events, assessments. It includes everything from subject screening through long-term follow-up. Everything is on this table, and we can sketch that out early. This helps us in solidifying our study design. So when we have an interventional study, what we want to do is we want to start with that synopsis. We want to start with that kind of skeletal frame of our study. We want to start having conversations and doing feasibility for our studies at this point. This is the perfect opportunity to loop in Dr. Marshall, if it's appropriate, to loop in lab, to loop in ancillary departments that may have input like radiology or allied health, audiology, neuropsych, physical therapy, anybody who has a role in the project, then we can loop them in at this point. We can start talking feasibility. Finalizing the synopsis and schedule of assessments prior to actually building your protocol is going to help you greatly in actually developing your protocol rather quickly. One note here is that our protocols, you see a lot of redundancy in our protocols. We talk about the same information multiple times in our protocols, and that's by nature just kind of the way that protocol development goes. We want to practice judicious redundancy, meaning that if we copy that same information multiple times over, if we amend that protocol, we're going to have to remember and go back and change all of those references. In your protocol, consider cross-referencing to one appropriate location. That way, when you make an amendment, you only have to change it once in the protocol, and we don't run the risk of overseeing one area, which can lead to confusion or lots of stipulations from the IRB. Okay, skipping over this slide, I think we all know what the title page is. So in our introduction, we want to have a background, rationale, and literature review for all of our studies. Now there are, upon occasion, a few studies that we can submit to the IRB that require very little in our background section. But I always tell folks, when we're writing our protocol, if we do it really well, we are well on our way to already drafting a solid manuscript. So in writing our background rationale and doing a thorough literature review, if that's solid, then we already have that piece of the manuscript for the most part written. Of course, you want to do the literature review to update once it comes time to write up your study. What this is going to do is provides the basis of the research. The IRB looks at this. They want to see that rationale. In fact, when we're filling out that application, we have to note the rationale for our research study. And then also we're going to talk goals in this section. We're going to define our objectives and specific aims. This is an area that I mentioned earlier is sometimes very difficult for our investigators. But we want our hypotheses or questions to be specific, simple, and developed a priori. The brief synopsis, I mentioned this earlier. So a brief synopsis is really like an abstract of your research paper. It should be no more than about 300 words because it's just like an abstract. And we're going to summarize the central elements. This should stand on its own. In fact, a lot of times when we're receiving invitations to participate in a protocol, what they will send the investigators is this brief synopsis. Quite frankly, a lot of times they'll send the synopsis, they're determining who's interested in participating, and they'll let us know that the final draft of the protocol is still in process. Endpoints. So another area that we really struggle with sometimes are endpoints. So our endpoints are identifiable changes that show the intervention did what it's supposed to do. So we want to have primary endpoints and secondary endpoints. These should be measurable, should be objective, and should be feasible. So I've provided a couple of examples over here for some rather vague objective endpoints and then some clear detailed objective endpoints. We want to make sure that our endpoints are referenced originally at the front of the protocol, and then we're also going to talk about them again when it comes to the analysis section so that we can specifically delineate how we're going to assess our endpoints. In the design section, we want to talk type of study design. As you know, in the protocol templates, we have some language that is pre-populated for some of our studies that makes developing the design language pretty easy. In the design, we want to talk about cost benefit, prevention, effectiveness. We want to talk about study population. We want to talk about study timeline. In our methods, we want to be detailed in describing our assessments with operational feasibility in mind, meaning that when we talk about study procedures, we want to be specific, but we don't want to be so specific that we're going to constantly deviate from our protocol. Recruitment, this is something that's come up recently and I think will continue to come up, which is under the recruitment section, we have to tell the IRB about our recruitment methods. So if we're going to have specific recruitment tools, if we're going to utilize advertisements, if we're going to put it on the web, if we're going to send out letters, we need to, in the protocol, reflect exactly what our strategies are. So consent, I'm skipping over this. We all know about consent. Off-study criteria. When we are working on developing an interventional trial, we really have to talk about that off-study criteria. So those things that will give us cause to remove a patient from a study. A lot of times it has to do with if sponsor or manufacturer is temporarily stopping drug or intervention. So Cook is no longer participating. Typically that's going to happen if some negative results have been published from manufacturer and we were just cross-referencing that study. Dose modifications or non-compliance. Those are 3 strong ones for when we would withdraw a subject. Our off-study criteria is going to be those conditions for which we would withdraw someone. Sources of research material. So in the sources of research material, you may have noticed that the IRB is getting a little more particular and asking some directed questions about where information is coming from. Typically, we say they're coming from the medical records, from medical data, and we kind of leave it open. If you are working on a study and we're pulling information from a registry, from a quality database, let's go ahead and be specific about that. So we're talking interventional study here. One of the big sections is going to be our investigational agent device, if it is a treatment study. So we're going to have the names, generic names, chemical formula, source pharmacy, supplier toxicity guidelines for administration, identification numbers. For the interventional studies that I have written, I will tell you, I spend a lot of time here. Where typically I'm looking on the FDA website, I'm pulling approval letters, I'm pulling package inserts, pulling as much information as I can find. If we're working with a manufacturer and they are willing to share their investigator brochures, and that's phenomenal, and you can find most or all of this information here. Not everyone that we have worked with has been willing to do that. Methods, study procedures. So again, we're just going to spell out exactly what we're going to do. We don't want to make it so specific that we're going to deviate. But while we're developing our protocol, we want to anticipate some common questions that could arise. So, for example, for vital signs, should the subject be sitting or supine? What assessments are meant by vital sign measures? An example of level of detail. So, too specific, if we write into our protocol that we're administering a drug at 0800 day 7, we are going to deviate probably 95% of the time. If we just say subjects will return for a follow-up visit, we're not going to deviate, but our data are going to be scattered and the IRB is going to step that back. What we want are just to add some windows in. So day 7 plus or minus 3 days, or whatever is an acceptable window. We want to include acceptable ranges. So as you may know, different labs often have different reference ranges. So when we are delineating lab values in our protocols, we want to be sure to include the acceptable ranges. And then state when procedures are optionals, of course. So our schedule of visits and assessments, we talked about that. This is what that table looks like. This was from, like I said, a expanded access study that we did on triheptanoin. And so it spells out pretty much anything that happens with the patient. Also, before we get started, Jeff uses this table for budget development and also the rainbow page build out. So you're going to take your schedule of events, you're going to set with your investigator and mark up which of these are standard of care. They'd be doing it regardless of whether or not the subject is participating in the research, and which of these are research costs. Standard of care is going to be built out and get billed to their insurance. Research is going to get billed to the research department to be paid for either by RAO or by your grant. So study procedures, safety assessments is incredibly important in interventional studies. So our IRB requires that our interventional studies have a DSMB, Data Safety Monitoring Board, or a independent medical monitor, or some other safety board. When we submit our protocols, we're going to have to submit information related to charters, related to who's monitoring, how they're monitoring, how often they're meeting, and so forth. That should be spelled out in your protocol. It doesn't have to be spelled out in detail, but it should be spelled out in reference to the charter so that if those individuals change, you don't have to go back and change your protocol. Study procedures, that adverse event section, many of you have seen and probably not had to use because we don't do a lot of studies with adverse events. I have written some templated language in there that hopefully works for most of the studies. There are times where we have to go in there, we have to decide our grading criteria and we have to change that. If you want just kind of standard language, that language is already in the template. Stopping roles. And then statistical considerations. So especially when we're writing interventional studies, we want to be pretty specific with our statistical considerations and plans in the protocols. So these sections can get a good bit lengthy. We want to talk about, with each of our aims, what analysis we're going to do to meet those aims. And we also, in this section, want to talk about selection of subjects. So did we power our study? And if we did, we want that write-up reflected in the protocol. And finally, quality control and assurance. So we're going to write into our statistical considerations how we're going to assure quality, how we're going to disseminate or report results. So we want to, in our protocol, reflect if we're notifying participants of their individual results, and then how we're going to disseminate those results, kind of what our end game is for the results.